So today's class, the last four classes are focusing on the what are the final applications of the various techniques and methods that you have studied in this particular course. So, like we have studied, how do you do cloning, PCR, variations of PCR, and hybridization, looking for a particular gene, how to measure transcript levels, how to study promoter activity, then how do you do sequencing? How do you design oligonucleotides? How do you label oligonucleotides? Can you have a radioactive labeling, non-radioactive labeling? And so many different techniques we have studied in this whole course. And now what I want to tell you, and also very nicely put up in the chapters in TA Brown, that what are the applications of this particular technology in the field of medicine. So we'll be telling you certain examples. And also in my next two classes, it will be applications of this technology in the field of agriculture. So I hope you can understand that when I'm talking about the applications and I'm talking about largely it is in the field of diagnostics or in therapeutics in the field of medicine. And of course, these methods have also been used in understanding the basic understanding of various mechanisms and pathways that are present or prevalent in any organism that we are studying. So how many so I think that most of the students that we have in the class today or who generally attend the class, they are they are PhD or MSR students. So how many of you are working in the field of medicine, any aspect of medicine? Am I audible?
Yes, ma'am.
Yes, ma'am. तो कितने लोग हो तुम में से जो how many PhD students are working in the field of medicine? I think students who might have joined Professor Sundar's lab. Anybody here? Or Professor Ishan Gupta or myself? Nobody is there. So you all are working in some microbe engineering or bioprocess engineering. I'm not getting any responses. How is it possible? Okay, how many of you are working in the field of agriculture? I guess none. And bioprocess engineering? How many students are there? Anuj, you don't want to tell? You will be working in the field of medicine.
Yes, ma'am.
So what happened? सब सब चुप क्यों हो? Oh God, इतने चुप मत रहो कि somebody gets scared. And what could be the reason? Anyway, with this exciting response, let me just go ahead with the class. Uh, so coming to the various applications, so I told that majority they are in the field of diagnostics or therapeutics. So I'll be telling you certain examples and certain techniques which are involved in diagnosis or any of these things that I mentioned here. So starting with diagnosis, so for diagnosis, PCR technique has been very, very valuable. And it is our knowledge both of the restriction enzymes as well as the PCR that yes, you can detect a particular gene by using a set of specific primers. It is just your intelligence how you design the primers that you can diagnose a particular altered gene versus a normal gene. So let's say here the example is given of sickle cell anemia, and I think we all know that it happens due to a single mutation, single base mutation. So how the various diagnostic assays they can detect that single mutation. So incidentally, in this case, it was realized that the mutation that takes place that lies within the recognition sequence of a particular restriction enzyme. So in this case, it is CBN1 enzyme. So in a normal sequence for that particular gene, you will get 4 fragments if you digest it with CBN1. And in case of a normal, in case of the disease gene in sickle cell anemia, then what will happen, because there was a mutation in the recognition site of a restriction enzyme, so obviously the mutation is there, so restriction enzyme cannot bind to it and this site is lost. So in this case, ideally we should see 3 bands. So if you have to diagnose individuals that, okay, how many of the population or people who have come to the clinic, how many of them have sickle cell anemia, how many are carriers and how many are have completely normal phenotype, both alleles, wild type alleles, then how do you do it? You first perform primer using a set of primers which is specific for that particular gene, and the PCR product that you get, you cut it with CBN1. So in individuals where the wild type allele is there, both wild type alleles are there, then you will get these 4 bands. I think there is a misprint in this particular figure. It should be into 10. I mean, it should be 380 base pair, 250 and 200 and so on. So anyway, so in this case, you will see 4 bands like I told you here. In case of sickle cell anemia, these bands will be absent. Rather, we'll have another band which is a combination of these 2 bands because there was loss of restriction enzyme site. So we'll get 3 bands. While those individuals which are carriers, that is, one allele has mutation while another allele is wild type, you will see a combination of these bands. So then you can designate that, okay, this person is carrier, this is suffering, both the alleles are sickle cell anemia alleles. Okay. Now, in this case, there was an advantage that by chance where the SNP was there or a mutation was there, that was the site for restriction enzyme. But obviously, in majority of the cases, it's purely luck basis that you may have a restriction enzyme site, but in majority of cases, you will not have a restriction enzyme site. So how do you look for single nucleotide polymorphism in these cases using PCR? So in this context, a very sophisticated technique was developed. It's called PCR-OLA, where OLA stands for oligonucleotide ligation assay. So let's say that we are looking for that in one case, I mean, in a normal or wild type allele, you have T at this position. While in case of the mutated DNA, you have a G at this position. Now I somehow have to identify that whether a mutant is present or a normal allele is present. So what they have done is that for this particular region, this spanning this region basically, they have designed 2 probes. So one probe, it ends in A, which is like corresponding to the wild type allele, and another probe they have designed which is from the other end of this particular mutation or the region that we are interested in, and it ends in the normal this base C, which is just next to the base that we are talking about. Okay. So what will happen that when I take out my DNA and I incubate it with these probes by hybridization, then this will bind here and this will bind here. However, if it was mutant DNA, then A will be hanging around and you will not see that it binds here, right? So, I mean, obviously, because it is not complementary to G. So, then what happens that after this step, once you do this probe attachment or hybridization thing, then you perform ligation using the enzyme called ligase. So what will happen that in this case, because A was bound here, you will get a phosphodiester bond that is made between A and C. While in this case, where you don't have the right nucleotide, then there will be no ligation, because anyway this was hanging here. It was not bound to this particular nucleotide. Now, how do I detect that whether my whole probe has bound to it, or the ligation did not take place? I mean, how do I find that the ligated product? How do I detect it? So for that, another very sophisticated method they thought about. In this case, this probe uh, where the end nucleotide was was the nucleotide that we were thinking about, that we are interested in, it has a biotin at one end. It is labeled with biotin. While the other probe from the 3' end, it is having digoxygen on this particular end. So what will happen that uh, once you are done with the ligation, then you pass these ligated products in a multi-well plate, and in each plate there is streptavidin present. Okay, so what will happen that because biotin binds very very strongly to streptavidin, so the all the biotin probes which are present, this ligated product, they will bind to it. So this will also bind to it, and this will also bind to it. Then I want to know that among the biotin labeled, these whole oligonucleotides or the ligated products, which one is having digoxygen at its end? So what do I do is, see, in this case, it will be this whole thing, while in this case, it will be only up to here, because no ligation took place. And anyway, after this ligation, I have denatured my DNA. I denature my DNA, so what will happen? This ligated product will be floating, and this particular strand will be floating. Similarly, in this case, when you denature it, this will be separate, this below strand, this will be separate, and this will be separate. Okay, so now then I treat it with, I put it in a well which has streptavidin. So this will bind and this will bind, and all the unbound products, they will be washed off. So what will be washed off? This will be washed off, this will be washed off, and this will be washed off in that case, because this was not bound to it. Now, how do I detect that, okay, in which well? Because I told you that in molecular biology, you can't see that, okay, yeah, dioxygen is bound here while it's not bound here. So you need to have either fluorimetric or colorimetric reaction to detect it. So how do you detect it? You use anti-digoxygen antibodies. So those antibodies, they can be fluorescently labeled, or you can add, secondary antibodies which are either fluorescently labeled, and you directly see the fluorescence, or you can use the antibodies which are bound to an enzyme, which in presence of substrate either give you fluorescent reaction, or they give you, chemiluminescent reaction, or they give a colorimetric reaction. So these anti-digoxygen antibodies, they have alkaline phosphatase or HRP bound to it. So these are the enzymes which in presence of a particular substrate will either give you color or a chemiluminescent signal. So this is how you can finally figure out that from the wells where you are seeing, let's say, purple color, if I have given some colorimetric reaction, those are the ones where the wild type allele is there, while the wells where the wild type allele was not there, I will not see any color or fluorescence. I hope, is this technique clear? Are you all sleeping? Is this technique clear?
Yes, ma'am.
Thank you for saying yes.
Yes, ma'am.
Okay. Anyway, the description is given in this particular slide, and you can go through it. But at the same time, I want to tell you that how sophisticated these are. Simple. The concepts are simple. You know the concept of hybridization, and you know that phosphodiester bond is made between two adjacent nucleotides. These simple facts, which you are aware of, that how they have utilized to develop a very sophisticated technique to detect the SNPs, and it's a very very sensitive reaction and highly specific in nature, and which is very much required when you are looking for SNPs in a whole population, and then trying to figure out that okay, this person maybe is prone for this particular disease, this person is not prone, and so on. So yeah, so these two methods are there, and one variation of this particular method for SNP detection is shown in this particular slide. So this is that how you can genotype with the fluorescently labeled primers. So in this case, what they have done, they have designed three set of primers. One is ending in the nucleotide, which is the right nucleotide that you are aware of, and the other one is ending on a nucleotide, which is the SNP that is generally found in that population. Like I would say that in one case it you see, generally you see a T, while in another case you see a G. So in other case means you design another primer which ends in a C, and one primer which ends in a A, and the 3' primer means the reverse primer, it is ending to the nucleotide which is next to T. So that is common for both of these things. Only thing is that in this case, what they have done smartly, they have designed these primers which have fluorescent probes. I mean, which are fluorescently labeled. Okay. So what will happen that in the case where you have the right or wild type nucleotide here, then again this ligation will take place and you'll get this whole thing. I mean, this whole product is there. Then what you do is that you want to detect the presence of the ligated product by the method of PCR. So if it is, in this case, if it is not ligated in between, and you perform PCR from the ends of this ligated product, then you will not get a band. However, if it is properly ligated and you perform PCR from the ends of the this particular ligated product, then you will get a particular fragment of a particular size. And also in this case, because you're using labeled primers, you can see red fluorescence. Now, how do you perform PCR? Which type of primers do you go for? So in this case, what they have done, again, smart thing, that they have added the M13 forward and reverse primer on either side. So you know that M13 forward and reverse primer means they are not present in the genome. So if you perform the PCR after ligation and all those things that I told you, using M13 forward and reverse primer, then it will only amplify this particular product. So what will I get is the product of this big size for a ligated product. So if I finally see a red color, I can assume that okay, in the original DNA at this particular position, the T nucleotide was there. If I see a green fluorescence, then I am sure that okay, in the in the well or the tube where I've done my PCR, the DNA, it was GG. Both the alleles were G and G. While in those cases where I get a yellow fluorescence, it is likely because I get signal from this also and I get a signal from this also, the mutated and the wild type. So it means it is present in a heterozygous condition. One allele has a G at one position while one allele has a T. So this is a variation of the previous technique, because in this case you had to do some extra steps which were little bit more cumbersome. You had to first attach it with streptavidin. I mean, you had to pass it through a well with streptavidin and then do immunoprobing using specific antibodies against digoxin. But in this case, it is comparatively easy. You can just do PCR followed by hybridization and ligation reactions, and then you can do PCR. Then another in diagnostics, when we talk about, obviously it is not always that you want to detect only the DNA. Many times you want to detect RNA. The example given here is for RNA viruses, but it's not obviously limited to that. Many times for disease conditions and all, you want to detect specific transcript, the changes in the level of specific transcripts, and for that I told you how do you design primers to detect those transcripts. How do you ensure that those amplification is only taking place from RNA, not from DNA. Then you can also design primers so that you only look for the alternatively spliced forms. So as we all know that in a particular condition, in a disease condition, there is a possibility that you get a different alternatively spliced transcript as compared to in a non-diseased condition. So even for those cases, all these cases, you need to know this reverse transcription followed by PCR. And this technique becomes very, very important when you want to see transcript changes. So in this case, you have to look for obviously coronavirus, the presence of it. So what you have to do is you have to design primers which are specific for any organism. I'm saying for any infectious organism. It could be corona, it could be dengue virus, it could be chikungunya virus or whatever. And what would be the source of the RNA that you get? It could be blood, it could be swabs, depending on which disease we are talking about. Many times people go for saliva, etc., etc., because you can detect RNA in various places. And only thing is, like I told you, you have to, whenever you have to isolate DNA or RNA, you have to take various precautions. Now coming to the PCR, which detects these infectious organisms, here you have to be very, very smart in designing the primers. Like in case of the RT-PCR kit that was developed at IIT Delhi, the only, see, everybody does RNA isolation and followed by PCR to detect a particular transcript. Only thing is how smartly you design your primers. So in this case, they compared the genome of various viruses and then finally came to a region which was very, very specific for this particular virus, and then designed primers, specific primers against it, and then just use the RT-PCR kit that is available in the market. So it is a simple thought. Only thing is that how smart you are in designing primers. Maybe in this case I'm talking about coronavirus, but in many diseases you can come up with a panel of genes or transcripts, their detection, by which you can very, very nicely compare the disease versus the normal state. So all these techniques you have done, I'm just saying that in which different scenarios you can actually apply it. Then DNA fingerprinting, I think that many of you might be aware of it, and it is generally talked about in terms of forensics, when you have to identify that the DNA which is present at a particular place, with to which individual does it belong to. So how can we, just by looking at the DNA, identify whether it belongs to the same individual or different individuals? Is due to the fact that in the genome of all the individuals, there are presence of various repeat regions. So there are various types of repeat regions. There are repeats which are present in tandem. Tandem means one after another, one after another. Then there are these repeats, some are very small in size, like 3 or 4 base pair, which are repeated multiple times. Then there are some which are 100, 200 base pair which are repeated multiple times. So various sizes of repeats and various these repeats which are present in tandem. I mean one after another. So these are called as the ones that I'm talking right now is about short tandem repeats, means short sequences of nucleotides that are repeated many times in tandem. So the thing is that in one individual, you may see that a particular STR region is repeated 3 times. In another individual, we may see that it is repeated 5 times. In some other, maybe 100 times. So it varies depending on individual to individual. Then how do you see that a particular STR is present how many times and all? So what you can utilize two different methods to see that how many times it's repeated by identifying the restriction enzyme sites on either side of these tandem repeats. So if I find, let's say, eco-carbon site which is present on either side, so I will isolate my total DNA, digest it with the eco-carbon, run it on gel, and then do probing against this particular STR. So then I can get to know the pattern of this particular STR in a particular individual. Another option is that I design primers from either side of these tandem repeats, the ones shown in blue, and then do PCR and then run it on gel. So again, for a particular individual, I'll get a set of fragments, PCR amplicons, which are specific for that particular individual. And then I can compare this pattern with pattern from another individual, and then say that okay, this belongs to this individual while this belongs to the other individual. Now there is a possibility that for a particular STR, in two different individuals we see exactly same pattern. There is a possibility for at least any one particular STR. You need to know that in the whole genome, there are multiple hundreds of thousands of different STRs which are present, or BNTRs. I told you there are different types of repeats. So that's why when you have to compare two different individuals, you go for a set of STRs which are present. Like I know last when I had seen that a panel of 13 different VNTRs or STRs, they were approved by the certain agency in US as a panel to compare between two different individuals. So what I want to say that ठीक है, किसी एक का पैटर्न फिर भी थोड़ा बहुत सेम हो सकता है, बट इट इज नॉट पॉसिबल दैट इफ यू आर प्रोबिंग फॉर 12 or 13 different STRs, that all of these will vary. So that's why what I want to say that a panel is generally seen, and for identification for a particular individual. So this is about DNA fingerprinting again, as you know. So means I hope, see, obviously I cannot cover every aspect of diagnostics, but in diagnostics, the concept of hybridization, the technique of PCR, the technique of restriction enzymes, these become very, very important. And another thing is you can also use, sequencing of amplified gene and see that what are the differences between the control and the disease sample, etc. Now coming to therapeutics, and obviously there is like immense contribution of RDT in the field of therapeutics. Nobody, I mean, we have come at that stage that we can acknowledge the importance of RDT, the cloning, the method of cloning foreign gene, and then amplifying it in various organisms for, and then harvesting it for therapeutic purposes is what this recombinant pharmaceuticals is all about. And there are several success stories. There are so many, just very small list is given here, but obviously we know there is a whole list of proteins which have been made in various organisms. Many are made in single cell organisms like E. coli or yeast, or various cell lines. in which these recombinant proteins are expressed and finally harvested. And it's not only limited to these cell lines or these single cells, but there are also work going on in which you can use whole transgenic plants or animals to uh, uh, to make these recombinant proteins and finally harvesting and purifying it. Because obviously if you are able to clone a particular gene in multicellular organism, then its amount will be humongous as compared to what you are making from E. coli. So you can harvest more protein. Obviously the development of these transgenic animals and plants will be more um, it will require a lot of effort and all, but once they are made, you can harvest a much more amount of these proteins. So currently in the market, what we see that majority of the therapeutic proteins that are available in the market, they are derived from mammalian cell lines, and the reason being simply being that for these proteins which are used for at least for therapeutic purposes. I'm not talking about the enzymes which are used in vitro reactions for doing a particular reaction, but the therapeutic proteins, they would require that they should be very similar to the proteins which are present in humans. They should be very similar. There should be minimal dissimilarity. Post-translational modification should be similar. So in those cases, you will go for mammalian cell lines because E. coli has not been efficient in doing post-translational modifications. But does that mean that E. coli and yeast, that market is completely off? No, obviously, because not all the proteins would require post-translational modifications. Many proteins which are expressed in yeast, which have some post-translational modifications. So even in yeast also you see that, okay, good enough protein with those properties are being made. And in E. coli, where post-translational modification is not required, you can get those proteins and do the post-transcriptional modifications later in a tube. So still this aspect is not gone of making therapeutic proteins in prokaryotic organism or in yeast. Uh, I have discussed before also that many changes they are doing in the host strains, be it E. coli or yeast, or they are looking for alternate host strains. I told you that initially it was only Saccharomyces that were used, but later it was realized that Saccharomyces is better. It's Pichia pastoris. Then it's realized that okay, Pichia pastoris is very good, but they can be better strains as well. So now people are trying to see that how many different strains means which strains can give uh, the therapeutic protein both quantity as well as its quality similar to the one present in humans. So it's a very dynamic field again, and people are trying to harvest these proteins from these sources. Also, I want to tell you that why we still want to keep these sources alive is the fact that growing the mammalian cell lines and maintaining them, it is very very expensive, and it is obviously time consuming as compared to if you are able to harvest it in E. coli or yeast. They, that how simply bacteria grows just in LB and all, but in mammalian cell lines, if somebody who comes to my lab, they will see how difficult it is to just to maintain these cell lines. Forget molecular biology, just to maintain these cell lines, and how expensive their media is, very very expensive. So when we talk in terms of industry, obviously we would want cheap source. I mean, kam lagao zyada power types. So E. coli and yeast are, that's why very very important. And then these insect cell lines are also being used. There is some work still going on with respect of transgenic plants or animals as source of recombinant proteins, which I'll discuss in my next classes. So we have several success stories. All these proteins which are mentioned here, just a subset, small subset of which are actually in the market. They are already being given to the patients for treatment. So I'll tell you just two examples of these, just to sensitize you that how this whole thing comes up. Each protein that I've mentioned here, it has its own story. Many of these proteins, they were first made in E. coli. Then it was realized, oh, it's not good. Then they went to yeast. Oh, it's good. Then they realized, oh, in yeast the quality is like this. Post-translational modification is there, but you need, comparatively better post-translational modifications. Then they go to another source, and so on. So each protein which is mentioned here had its own long story of development. And just two examples I'll come to. Obviously, I cannot ignore insulin, which has been the major success story, especially because insulin is so much used in the market. I'm sure that every one of you must be having 4 5 relatives or friends who are already suffering from diabetes. It's not only now prevalent in adults, in young children also now you see diabetes, and majority of these they are being treated by giving insulin. So you can understand how huge market is for insulin, and the proteins or various therapeutic proteins which for the diseases which are very prevalent in our population. So insulin, it is first expressed as this whole, this is the translated product. So it has a leader region followed by B, C and A chains. Then leader is removed, and what we get is a proinsulin, which is BCA with no leader. Then once you get this proinsulin, then finally this C chain is removed, and it is B and A that come together to make the final insulin. Now, the thing is that how insulin was overexpressed for and finally, harvested for therapeutic purposes. So initially, they had two different plasmids, and in each, in one case, they thought that okay, see, only A and B is finally used. So why to clone C? So they just in one they cloned A, in one they cloned B and harvested the protein. And because this A and B protein, they were part of, means it was in MCS, and it was realized that a small part of beta galactosidase was part of the final chain. So it was realized that okay, because we want to use it for therapeutic purposes, this particular short chain has to be removed. Now, if you remember, I told you that there are various ways by which you can remove these fusion tags, and one of which is that if there is no, methionine present in the body of the protein, then if there is only one methionine at its start, then you can treat it with cyanogen bromide, which will remove everything. Cut at methionine, and this extra polypeptide, it will be removed. So then we get this pure A and B chains, and then you combine them in a tube and you perform a reaction for formation for disulfide bridges. So this is how initially this insulin was made. So now currently the insulin which is in the market, some is many are from E. coli source, while some companies they are making it in yeast. And some it was realized that can we have A and B chains together in one vector. So in many companies, they are making A and B in one vector only. And then in some companies, they are thinking that why to, have A and B chains, like I told, separately. So in those cases, they are expressing proinsulin. So they have a cDNA which codes for just proinsulin without the leader region, and then making insulin. And in many cases, what was realized that insulin, it is not, once you standardize it that okay, my job is done, now insulin can be easily made in the market, in the lab, and then sold to the market. Then it was realized that insulin, there are two types. One is slow acting and one is fast acting. So, and if you think about in the terms of clinics, Many times, the doctors, they want fast acting insulin. Why? Because if the sugar has really shot up, they want to treat the patient immediately with fast acting insulin, and they don't want to go for slow acting. And in many cases, the patients are treated with slow acting, and in many cases it's a combination of these two types of insulin that is prescribed to the patients. So it was realized that when you this insulin is harvested, usually it was found in oligomeric form, and this are not harvested, sorry, when it was delivered to the patient. So it was found that it is present in oligomers, and because it was present in oligomers and bound to certain proteins, it became generally slow acting in nature. So then there was, lot of discussion how to prevent its oligomerization. Is it possible to just ensure that it is present in monomeric forms? So then they try to look that what is the reason for presence, for its, combining together in oligomeric forms. Then they realize that it is the presence of certain side chains in certain amino acids that is promoting the oligomerization. So that's why I told that basic biology as well as, applied biology has to go hand in hand. It is only if you realize that what amino acid is responsible for a particular phenotype that you can change it. So once they identified that okay, these particular amino acids, they are important for oligomerization, so then they fiddled with the side chain, or they changed those amino acids. So in various experiments, some people have changed some amino acid, and some they went for a combination of amino acids, and some they went for totally different amino acids. And now we do have the insulin, which does not oligomerize and just stays in monomeric forms, and that is the fast acting form. So insulin, this story, it is very very interesting, and you can understand the dynamism, to the fact that now people are trying to see that if you can make insulin in the plants, you use plant products, I mean plant parts, as the source for making insulin. So there are studies going on in which they are trying to overexpress insulin in the seeds of the plant. So and they are also, trying different organisms for making insulin. So still a very very well established protein, the overexpression of which has been very successful. Still experiments, various industries, there is a lot of research going on how to improve it further and further and so on. So I have in the notes section included a paper which tells you about the journey of insulin. I found it really, really very interesting. So I would suggest that please go through that paper just for your own understanding that how a particular field is developed over time. This is the another example that I want to give you. It is about the overexpression of recombinant factor 8, this particular clotting factor, which is used for the treatment of diseases, especially hemophilia. So this is a very, very big gene. So obviously it is the cDNA that was finally cloned for making this particular protein. So in this case, we have these A, B, and C, these three different chains. In the final product, the B is removed and it is only A and C which are joined together, and it requires the presence of multiple disulfide bonds. So in this, what they did was that they cloned the whole cDNA, and they introduced it in hamster cells, but they realized that the yield of protein was very low, very, very low, even though the whole cDNA was there. So now what they have done is that they have expressed A and C separately, and then they are making this recombinant protein. So why I am telling you this example is only for introducing you to one fact that that you have to, try different methods to get the optimum amount of a particular protein that you are interested in. And second is that the promoter, look at the promoter that they use. It's something called as AG promoter. So, in the AG promoter, it is a hybrid between beta actin and beta globin sequences. So, why I'm telling you this is that, see, one thing is you can be nostalgic that yes, there was something like that about a prokaryotic promoter also. If you remember, I told you about that TAC promoter, which was a hybrid between lactose and tryptophan promoter. And what they found that the yield of a particular protein which is expressed downstream of TAC promoter was much higher as compared to LAC or tryptophan separately. So similarly, they have developed a promoter which is a combination of beta actin and beta globin genes. So here I want to stress to the fact that it is not only, the strain that is important, but obviously it is the promoter that are very, very important. And many of you can actually think about developing various promoters. There is a need for promoters which are very strong promoters as compared to the already existing promoters. You can try to make mutations in those and see if you can get a better promoter. You can try to combine two different promoters to make a hybrid promoter, or you can make many times there is a need in the market to have tissue specific promoter, that only these products will be expressed in only a particular part of that organism. It could be that you only want a seed specific promoter. You only want a very strong promoter which is only expressed in the leaves. There is no need for it to be expressed in other parts of the plant, because maybe you are only interested in harvesting the leaves of that particular plant. So there is a need for all these things in the market, both for basic research as well as for applied research. So you can think about, not only just over expressing a protein, but developing the promoters or the very strong polyadenylation signals. So as molecular biologists, you have to be extremely observant of the things that you're working upon, and think of the ideas how you can make changes in these and come up with a some tool that can be very useful for the researchers. Then yeah, so in this case what they had done means, why I have shown you this example is purely for you to think about that many changes can be done. Also, I forgot to mention, obviously you also need a very strong inducible promoter. So if you remember how those pet vectors were made with so much elegance, they thought about that, okay, let's make introduce this T7 RNA polymerase and make it lactose inducible and so on. And how did they, to ensure that the expression was not too much, they added P lyse S, that particular thing. If I don't know if you remember or not, but yes, all these things you should think about. Be very observant and keep your minds open and try to develop something good as part of recombinant DNA biology or molecular biologists that you are. So that is all for today's class, and in on Friday I'll be continuing with this the scope of applications of this recombinant DNA technology in the field of medicine, and see you on Friday. If there are any questions, you please ask now. Okay, so if there are no questions again, then have a